Researchers at the University of Illinois Chicago have developed a unique method for precisely controlling the deposition of hydrogel, which is made of water-soluble polymers commonly used to support cells in experiments or for therapeutic purposes. Hydrogel mimics the extracellular matrix, the natural environment of cells in the body. The researchers noticed that their technique, which allows for the encapsulation of a single cell within a minute hydrogel droplet, can be used to coax bone marrow stem cells into specialized cells. Their findings are reported in the journal Advanced Science. The new technique is an improvement over existing approaches that often mix much larger amounts of hydrogel with cells in an uncontrolled manner, which can make interactions between cells and their surroundings difficult to study. The new hydrogel deposition technique may also be useful for therapeutic purposes, such as for supporting stem cells used to create new tissues. Most experiments use a very high amount of hydrogels to interface with cells, which may not reflect what is happening in the body, said UIC's Jae-Won Shin, assistant professor of pharmacology and regenerative medicine at the College of Medicine, and assistant professor of bioengineering at the Co. According to Shin, the team's deposition technique brings the ratio between hydrogels and cells in line with what is seen in the body, and importantly, precisely controls the ratio on a single-cell basis. Shin and colleagues also observed that stem cells in thinner gel droplets expanded more rapidly than they did in bulk gels. We observed that stem cells expand several orders of magnitude faster in thin gel droplets, and so they experience more tension than they do in bulk gels made of the same material, said Seung-Won Wong, a postdoctoral fellow in Shin's lab and first author on the study. We believe this tension encourages stem cells in thin gel coatings to more readily become bone cells, compared to stem cells in bulk gels. The team believes the thin hydrogel deposition technique may help in the production of bone tissue from stem cells to use as regenerative therapeutics. Stephen Lenzini, Raymond Bargy, Celine Makaraniag, James C. Lee and Zhangli Peng of UIC and Zhefeng of the University of Notre Dame are co-authors on the paper. This research was supported by grants from the National Institutes of Health, RO1HL141255, ROOHL125884, and the National Science Foundation 1948347 C.B.E.T. Story source: materials provided by University of Illinois at Chicago. Note: content may be edited for style and length. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you new tech videos. Thank you.